Welcome students. This is for the fourth sem students, tissue culture. Anyways, this is the lecture number 2. Already last class we have seen the definition for plant tissue culture and a history and about types of cultures. Today we are going to discuss about the nutritional requirements, growth and their maintenance. Nutritional, that means nutrient media for plant tissue cultures. One of the most commonly used media for plant tissue culture is that developed by Morashik and Skog. MS media. You can say in books they used to say MS media. First it was developed for tobacco tissue culture. First we should know functions of media. A media's function, first one's to provide water, provide mineral, nutritional needs, provide vitamins, provide growth regulators, and access to atmosphere for gas exchange, removal of plant metabolite waste. These are all function of media which will be provided for the tissue culture. Components of MS media. When we talk about components, number 1, inorganic salts, carbon sources, vitamins, growth regulators, organic supplements, and media for plant tissue and cell cultures. We are going to talk about what all the inorganic salts is the components added. Inorganic nutrients, it includes mineral salts that are important for the growth and development of plants. It is categorized into 2 groups: macronutrients or calcium, magnesium, nitrogen, and micronutrients or nutrients, copper, iron and zinc. Carbon sources, sucrose or glucose at 2 to 4% are suitable carbon sources which are added to the basal medium. Fructose, maltose and other sugars also support the growth of various plant cells. An optional concentration should be chosen to establish the efficient production process of useful metabolites. See, vitamins. MS medium include myo-inositol, nicotinic acid, pyridoxine hydrochloride, thiamine hydrochloride. Among these vitamins, thiamine is an essential one for many plant cells, and other vitamins stimulate the growth of the cells in some cases. The next one is D, growth regulators. It includes auxin, cytokinin and gibberellins. It is essential for the growth and development of tissues and organs. E, Organic supplements. These are supplements include casein amino or casein. It is taken from casein. That is a casein amino acid, peptone, yeast extract, malt extract, and cocoa in order to stimulate the growth of cells. Organic supplements are sometimes added to the medium nut milk. Casamino acids are all of the natural occurring amino acids and particularly rich in essential amino acid. Cas amino acids have undergone a nearly complete acid hydrolysis of the casein protein until all the nitrogen is converted to free amino acids, intended for laboratory and manufacturing use only. Coconut milk is also known as a supplier of growth regulators. In order to stimulate the growth of the cells, organic supplements are sometimes added to the medium. Preparation of medium. To prepare the medium, many researchers mix the stock solutions which were made previously, since the medium composition are generally complicated. For example, MS medium is prepared as follows. MS micronutrient stock solution which are stored in freezer. A. Inorganic salts. Ingredients in milligram per ml is denoted. Boric acid 620 milligram, magnesium sulfate 2230 milligram, that means 2.23 grams, zinc sulfate 860 milligram, sodium molybdate 25 milligram, copper sulfate 2.5 milligram, cobalt chloride 2.5 milligram. Vitamins, they are also stored in freezer. Vitamins milligram per 100 ml, nicotinic acid 100 milligram, thiamine 1000 milligram, pyridoxine hydrochloride 100 milligram, myo-inositol 10,000 milligram, that means 10 grams. Calcium chloride separately prepared, calcium chloride 15 grams in 100 ml. Next solution, potassium iodide stored in amber colored in refrigerated because light sensitive. Potassium iodide, not chloride, potassium iodide 75 milligram in 100 ml. 2,4-dichlorophenoxyacetic acid, oxen derivative, plant hormone derivative, dissolved 50 milligram of 2,4-dichlorophenoxyacetic acid in 2 to 5 ml of ethanol. Heat slightly and gradually dilute to 100 ml with water. Store in refrigerator. 1 naphthyl acetic acid, it's also oxen derivative, plant hormone derivative. 50 grams of naphthalene acetic acid in 2 to 5 ml of ethanol. Heat slightly and gradually dilute to 100 ml with water and store in refrigerator. And kinetin, cytokinin derivative, dissolve 21 gram of kinetin in a small volume of 0.5 normal hydrochloric acid by heating slightly and gradually dilute to 100 ml with distilled water stored in refrigerator. Similar procedure can be used for other cytokinins if we are using in the medium. Other media substances which promote growth of the tissue cultures, like protein hydrolysate, that is example soya protein hydrolysate, yeast extract fruits, etc. Yeast extract fruit, example banana, and extracts coconut milk fresh pasteurized, and phenolic compounds like fluoroglucinol stimulates root and shoot section. Activated charcoal is used as an detoxifying agent, detoxifies waste from plant tissue and impurities. Absorption quality vary. Concentration normally used is 0.3% or lower. It absorbs the secondary products secreted by the culture tissue. Charcoal for tissue culture, acid wash and neutralize, never reused, controls the supply of endogenous growth hormones. Demineralized water is also can be used. Solidification agent which is used in this media are solid media are often preferred because it improves oxygen supply and support cultural growth. Therefore, substance with strong gelling capacity is added into the liquid media. The most common used substance is agar and other are alginate, carrageenan, gelatin, starch, silica gel, hydroxyethyl cellulose and polyacrylamide. Agar is extraordinary resistant to enzymatic hydrolysis at incubation temperature, and only few bacteria exist which are capable of producing degrading enzyme. Iron source, we are using EDTA, can be used as an iron source. pH of the medium should be in a range of 5.6 to 6.0 before autoclaving. The culture medium, that means before sterilizing the culture medium. Antibiotics are used for preventing the excessive contamination in the culture medium. Commonly used antibiotics are carbenicillin 500 milligram per liter and augmentin 250 milligram per liter. And next, a certain volume of each stock solution is mixed and an appropriate carbon source is added to the mixture. After the pH is adjusted to around 5.5 with 0.2 normal potassium hydroxide or 0.2 normal HCl, diluted or deionized water is added to the mixture up to the certain volume required. Agar usually 0.6 to 1% weight by volume is added for a solid medium. The medium thus prepared is distributed into vessels such as Elmer flask. For example, 50 ml of medium in a 300 ml volume Elmer flask. Elmer flask are available in different volumes and sterilized by using an autoclave at 120 degrees centigrade for 15 minutes. The sterilization condition should be varied based on the volume of the medium and the size of the vessel. This is Elmer flask 500 ml capacity which we have in our laboratory. Autoclave different sizes, room autoclave, big shelf autoclaves are also available. We can use according to the quantity which you are preparing. Equipments required. First one is laminar airflow cabinet. Autoclave, different types, different sizes are there. Oven for dry sterilization. Equipments for sterilization can be done by filtration as shown in the picture. Microcellulose membranes can also be used for filtration by means of this Buchanan funnel. And we can use water distillation apparatus and deionizing and also ionizing chromatography deionizing instruments can also be used for this purpose. Culture rooms or wide rooms are available where the culture can be kept, and also shelves with white lights, visible lights, which can be seen. Any contaminations which is visible should be discarded. And shakers, different types of shakers which we are using, like magnetic stirrer and rhythmic shakers, rotating shakers, centrifugers, these all are needed in different forms. The picture which is shown for you. Equipments required, especially first we are going to discuss about the small definitions. Laminar airflow cabinet, which you know. The cabinets are commercially available in different sizes. They are placed in laboratory where needed. If there is a sterile room, the cabinets are not always necessary. Autoclaves in different sizes are commercially available. Oven for dry sterilization. Although autoclaves can be used for dry sterilization, an oven is useful for sterilizing or sterilization of scalpels and glasswares such as petri dish, pipettes and others. Equipments for sterilization by filtration, as we shown in the picture. The medium contain carbon sources and growth regulators are simultaneously sterilized using an autoclave, but sometimes aseptic sterilization filtration is favorable to avoid decomposition of unstable chemicals due to thermal effect. The equipment is also commercially available. Number 5, water distillation apparatus or pure water demineralizer. To prepare media, distilled water and deionized water is generally used. Although tap water can also be used in some cases, particularly in large scale cultivation of plant cells in a large fermenter from the economical point of view. Number 6, cultural room or culture room and or cabinet. To cultivate the plant cells, culture rooms under different temperature or cabinet type incubators are essential facilities. Temperature and light intensity as well as duration of light in the room or in the cabinet are controlled under optimal conditions. Shelves built from rigid wire mesh to allow maximum air movement and minimum shade should be used in the culture room. Shakers, a rotate shakers and a reciprocal shakers is necessary for suspension cultures. Culture room of incubator will be like this: wider. The researchers can enter inside with a mask and a covering of a protection medias, and a room incubators are also available like shelves. Cultures are incubated on shelves or in incubator under specific condition of temperature, humidity, air circulation and light. Incubation chamber or area should be both light and temperature control devices managed for 24 hours period. Generally high output cool white fluorescent light is preferred for a photo period duration, specific period of total darkness as well as for higher intensity light with a temperature range of 25 plus or minus 2 degrees centigrade, range 18 to 25 degrees centigrade. The rooms are required to be maintained at a relative humidity up to 70 to 75 percentage, range of 20 to 90 percentage controllable to plus or minus 3 percentage, and uniform forced air circulation. Proper and optimum aeration. Adequate aeration is required for cell to grow. Tissue which are cultivated on semi-solid media do not require any special method for aeration, but tissue which are grown in suspension culture require special devices for aeration. Aeration for submerged culture can be provided by following methods. Place the culture vessel with a liquid medium on an automatic shaker. Two ends of a filter paper are dipped as shown in the picture in a medium, and the middle horizontal portion on which the tissue is placed remain above the level of the medium. This method is called as filter paper bridge method. Placing sterilized air through the medium and by stirring the medium, the culture vessels are closed with non-absorbent cotton covered in cheesecloth. This process also proper aeration allows proper aeration and prevents the entry of microorganism. So you should remember about the non-absorbent cotton covered in cheesecloth. Basic method or methodology technique of tissue culture, which you should remember: number 1, preparation of suitable nutrient medium, and number 2, selection of the explant, number 3, sterilization of explant, number 4, inoculation, transfer of the explant to the medium in aseptic manner, number 5, incubation, and number 6, regeneration, number 7, hardening, number 8, plantlet transfer to the normal greenhouse. So you should know these 8 steps for a basic technique of tissue culture to be performed, or basic methodology. So basic methodology, when we talk about the general technique used for isolation of growth of culture. Isolation and growth of culture. Preparation of suitable nutrient medium. As per selection of plant medium is autoclave. Selection of explant. Any that is excised part of the healthy plant to be used is decided, the bud, leaf or root or a seed, etc. Sterilization of the explant is needed by sodium hypochlorite, mercury chloride, etc. and wash aseptically for 6 to 10 times with sterile water or sterilized water. Inoculation transfer. The sterile explant is inoculated on a solid or solidified nutrient medium under aseptic condition. Incubation, the next step. Culture are incubated at 25 plus or minus 2 degrees centigrade at a relative humidity up to 50 to 70% for 16 hours of photo period. Regeneration. Plantlets generated after transferring a portion of callus into another medium. The induction of roots and shoots are directly from explants. Hardening is the gradual exposure of plantlets for accumulation or assimilations to environmental conditions. Plantlets are transferred to greenhouse or field conditions. So this is the picture which you can show how the procedure is followed. From a plant you are taking a washing with sterilized sodium hypochlorite, trimmed, and explant is prepared, trimmed and sterilized medium, washed with a distilled water or an sterile water, and it has been transferred to the medium or inoculation, final trimming and establish the culture as we shown. And next the callus is developed and subculturing into the different media as shown in the picture and incubating at even 25 plus or minus 2 degrees centigrade, 50 to 70% of relative humidity for 16 hours. You can see the buds are formed or shoots, micro shoots are formed, and the roots development and plantlets, and it can be transferred to the greenhouse and polythene cover, and it can be developed. And the same cycle can be repeated for developing another callus culture. So this is another example of picture to show you the selection of the plant in petri dish, how you are slicing, explant preparation, preparation of explant in a sterile condition in an aseptic manner, and inoculation. After inoculation, various stages of plant growth, and plant ready for transferring to the greenhouse by means of the regeneration of the plant from the explant takes place. And this can be done. And cultivation of the plant culture, some of the things, organogenesis. Organogenesis, if you see, explants, different explants, flower, leaf, root or stem, all organs can be developed and whole plants can be developed from the part of the explant. That is organogenesis. Organs are developed in the process involving redifferentiation of meristematic cells present in callus into shoot buds or roots or even whole plantlets. The formation of organ is called organogenesis. Embryogenesis. Embryo from the same explants. Embryo can be developed into a whole plant. Embryo is defined as an earliest recognizable multicellular stage of an individual that occur before the development of characteristic organs of the given species. Production of embryo-like structure from callus is known as embryogenesis. In higher plants, such embryos usually arise from zygote formation and are termed as zygotic embryos. Advantages of organogenesis and embryogenesis. 1. Efficiency of process, reduction in labor cost and time, the formation of plantlets in fewer steps, the potential for production of much higher number of plantlets, and morphological and cytological uniformity of plantlets. Production of several plants on commercial scale include food crops, vegetables, spice and fruits, medicinal and aromatic plants. Thank you for being with me. This is the second class. Next we will continue with the application of tissue culture in pharmacognosy. Thank you.